Can you hear me?
Yep, perfect.
Okay. Thank you so much for the invitation. So today I'm going to talk about antimicrobial peptide. I'm from Chapman University. For those of you who don't know, where is Chapman University is in California, Southern California, School of Pharmacy. So today I'm going to talk about antimicrobial resistance. This remains one of the major threats to global health, and we call it a silent pandemic. And if you know that COVID caused about 6 million deaths, so it's predicted by year 2050, we will have 10 million people death mortality every year because of antimicrobial resistance. Currently, we have about 2 million antimicrobial resistant infections every year in US and about 35,000 deaths. It's predicted that overall in globally is about 1.2 million deaths every year right now. So again, this is a major issue. And if you look at many of these antimicrobial resistant like vancomycin, methicillin-resistant bacteria, Staphylococcus, they're increasing year by year because many of antibiotics we have are not working. So in terms of major antibiotic resistance, we have SCAPI pathogen. We call them SCAPI because they stand for Enterococcus, MRSA, Actinobacter, Klebsiella, Pseudomonas, and E. coli. And as I said, MRSA is one of the major bacteria, and globally, we have about 100,000 deaths every year. In US, is about 11,000 deaths every year because methicillin doesn't work, and some of these superbugs are not reacting to any antibiotic. So, and if you look at the general cost of this, we have about $2.6 billion market. Unfortunately, a lot of pharmaceutical companies are not investing in antibiotics because, as you know, antibiotics are not profitable. And after the patient use antibiotic for a short time, they are treated. So they are not like anti-cancer drugs or antipsychotic drug that the patient have to use for a long time. So, but again, because this is a very emerging issue, I'm confident that many pharmaceutical companies eventually will jump into this because this silent pandemic is coming up. So in terms of antimicrobial peptide, there are more than 2,000 antimicrobial peptides have been identified from natural resources. We have sources like plant, and human also have an innate immune system that generates a lot of antimicrobial peptides, especially in our GI system. There are normal flora of bacteria that generate antibacterial peptide, and a lot of animals and insects. And usually the antimicrobial peptide, usually they are large, they are composed of 12 to 50 amino acids, and they are kind of hydrophobic and positively charged. Many of these are linear peptides composed of hydrophobic amino acid and positive charge amino acid. And the number of positive charge usually high, and the large number of amino acids cause that the peptide is not easy to be used in vivo, and also generate a lot of immune response because of presence of positive charge. But there are some advantages to antimicrobial peptide, especially in terms of broad spectrum activity. They can be active against gram-negative and gram-positive bacteria, and they have very rapid action. Some of them also have anti-cancer activity, and many of these bacteria generate aggregated biofilm structure that these antimicrobial peptides are acting against. They have a lower risk of resistance because they don't have a specific target. They usually work on the bacterial membrane, and it's very hard for the bacteria to develop resistance and change the membrane for a new antibiotic. Some of them have low toxicity and high selectivity. So, as I mentioned, they have many therapeutic applications, not only for systemic infection, but they can have activity also for cosmetic application, topical application, and anti-cancer agent. And they usually are synthesized by different technique and bioengineering technique. But then some of these techniques are very high cost, and we have poor metabolism, ADME property, and many of these peptides are very unstable. So therefore, they cannot be used orally. So that is some of the major disadvantages. So currently, we have several antimicrobial peptides in the clinical use. Polymyxin B, it has a peptide backbone, cyclic peptide backbone structure, and a very short fatty acid amide structure that this is derived from the bacteria. Vancomycin, that I'm sure you are familiar with, is a glycopeptide. It has a carbohydrate moiety attached to the peptide backbone. And then we have also daptomycin is a newer drug. Again, we have a peptide backbone, cyclic peptide, that mostly have a hydrophilic structure and attached to a side chain fatty acid. So as you can see, many of these peptides have amphiphilic property. So my research is designing cyclic peptide for multiple application, various application. So we designed some peptides that are alternate hydrophilic and hydrophobic amino acid. And as you know, tryptophan is the most hydrophobic amino acid. So we incorporated tryptophan in the backbone. And arginine, as you know, has a positive charge. So we incorporate it as hydrophilic amino acid. So when we incorporate this peptide as a alternate hydrophobic and hydrophilic amino acid in the cyclic structure, we kind of generate compound that has a very low toxicity, but it can be used as a drug delivery agent. We found out that this peptide actually generate a nanoparticle, and they can encapsulate with anti-cancer agent, antiviral agent for drug delivery. Or they can be conjugated with anti-cancer agent for drug delivery, and they are active against cancer-resistant drug, and they can deliver different anti-cancer agents. On the other hand, when you put all the hydrophobic group in one side and all the hydrophilic group in another side, like all the arginine in one side, all the tryptophan on another side, we generate a compound that actually has not only have some delivery property, but also has antibacterial agent. So for example, if you have 4 arginine and 4 tryptophan, this peptide generated significant antibiotic activity. So we call it R4W4. R stands for arginine and W for tryptophan. And we found that this peptide actually have MIC, minimum inhibitory concentration, about 2.67 against MESA, that is gram-positive, and about 42 microgram per mil against Pseudomonas, that's the gram-negative. And the mechanism of action, as based on our studies, shows that this peptide actually interacts with the bacterial cell membrane and generate pore inside the bacterial membrane and cause leakage. And so we're going to talk a little bit more about mechanism of action, but we hypothesize that arginine residue interact with the negatively charged bacterial membrane, phosphate group, and hydrophobic group interact with different lipids in the bacterial membrane. So based on this study, we came up with the idea, okay, if we have R4W4 that have hydrophobic residue and positive charge, You can design a peptide that have a positive charge amino acid on one side, and then a hydrophobic group in the other side. So as you can see, we can use different hydrophobic amino acid in one side of the peptide. And some of this work has been recently published. So the goal is to have this cyclic peptide that hydrophobic residue are like diphenylalanine, naphthylalanine, and biphenylalanine instead of having the tryptophan. So tryptophan will be replaced with this hydrophobic amino acid that are unnatural amino acid. And when we do that, we actually improve the stability of the peptide. And then also, because of the cyclic structure, the compound is more rigid compared to the linear peptide. We also get a more stability toward the peptides in the body. So if you are familiar with the peptide synthesis, we have resin, and we assemble different amino acids on the resin, so you can make using protection and deprotection, and then you can actually generate a linear peptide like this and cleave it from the resin. On the other hand, we can also cyclize the N-terminal with the C-terminal of peptide and generate a cyclic peptide like this, and incorporate many of these amino acids I told you, biphenylalanine, diphenylalanine, and naphthylalanine. So that is how we synthesize many of these peptides that include linear and cyclic peptides. So and we designed about 250 peptides and synthesized many of them, and just summarize some of the activity, structure-activity relationship. So if you look at the linear peptide, you can see that some of these peptides actually are quite potent against gram-positive and gram-negative. And then we also measure the hemolytic activity, the toxicity against the red blood cell. So as you can see, the peptides that have 3-arginine and 3-diphenylalanine or 3-arginine and 3-naphthylalanine show the activity around 6.2 to 2, around about against the gram-positive and around 25 gram-negative. Similarly, if you have 4-arginine, 3-diphenylalanine, 4-arginine, 3-naphthylalanine, we get a slightly better activity. If we use the cyclic peptide and we make the cyclic structure of the peptide, the activity actually increases, and we can see that, for example, the cyclic peptide, we have 3.1 to 12.5 against gram-negative and gram-positive. For similar corresponding peptide that when we compare with the peptide that was a linear peptide. And because of the activity of these 3 peptides, we call it 2C, HC, and 9C, they showed good activity. We tested them against many drug-resistant bacteria that are resistant, for example, to vancomycin. And we see the activity of this compound against the vancomycin-resistant gram-positive or daptomycin-resistant, or also many antibiotic carbapenem-resistant gram-negative bacteria. So that is showing that our compound actually are acting against multi-drug resistant gram-positive and gram-negative at low macromolar activity, microgram per mil, around 3.5, 3.1 to 12.5. So based on this bed comparison of 2C, HC, and 9C, we found out that HC and 9C are more potent than 2C. So we selected these 2 peptides for further study, and we did the bactericidal kinetic study of this peptide against different gram-positive and gram-negative bacteria. In this case, we have MRSA and we have also E. coli. So if you look at the ciprofloxacin and daptomycin compared with the control, and if you use time-dependent study, you can see that, for example, ciprofloxacin reduced the growth of MRSA and also E. coli, but it never eliminates completely. Similarly, daptomycin at higher concentration can completely eliminate, but at a short period of time, it cannot do that. And so these antibiotics are actually bacteriostatic. They are not bactericidal. They don't kill the bacteria completely. But when we look at the 8C and 9C for both MRSA and E. coli, we can see that first of all, the HC is more potent than 8L. That is a linear corresponding peptide. And they can completely eliminate the peptide after 2.5 hours. Almost all the bacteria are dead. So these compounds are actually bactericidal. They can completely eradicate peptide under similar condition that we use antibiotic. So if we look at the mechanistic study, so we design a liposome that has a dye, we call it calcain dye, and this liposome has a bacterial membrane. And also we design another liposome that has a normal cell, a mammalian cell membrane, and we also encapsulate the dye. So when we look at the incubation of this peptide with this liposome, if the peptide actually causes leakage in the liposome, we have the release of dye, so we can measure the leakage. So in case of daptomycin, we don't see much leakage, but in terms of HC and 9C, we see a lot of leakage of the dye from the liposome. So it means that the peptide actually generating leakage and causing membrane perturbation and change the fluidity of the membrane. And the way this peptide actually works, it binds to the membrane and it has general conformation. It binds to the membrane to the positive charge, and then it can flip inside the bacterial membrane, and this tryptophan residue can have interaction with the lipophilic chain in the bacterial membrane. And if we compare bacterial membrane mimicking liposome with the mammalian membrane mimicking liposome and look at the percentage of dye leakage, as I mentioned, for the daptomycin is very low, it's about 30% maximum. So if we look at the comparison of bacterial membrane, we can see complete 80–90% leakage at different concentration. In terms of the mammalian membrane, is less than 10% dye leakage. So this shows that these peptides actually are selective against the bacterial membrane, and we see a lot of selectivity because of the bacterial membrane are different from the mammalian membrane. They have much higher lipophilic, negatively charged lipophilic change that can interact with the positive charged peptide. And another study that we can do is that we can use the propidium iodide. Propidium iodide is a dye that actually interacts with the nucleus of the cell, and it can stain the nucleus of the cell. So if we have no treatment, so propidium iodide cannot get into the cell, and we don't have any stain cell. If we use a surfactant like tritone, we have 100% stain, because it means that propidium iodide can get into the nucleus. If you look at the antibiotic like daptomycin, polymyxin, we have slightly less than 20% for daptomycin and 40% for polymyxin dying of the nucleus. But in the presence of the peptide, we can see we have much higher staining of the nucleus. And also, if we increase the concentration of the peptide from 1 MIC to 4 MIC, even we get more staining. So that's another evidence that shows that the peptide actually interacting with the bacterial membrane and causing the leakage. So if we look at the electron microscopy study of the bacteria, when we don't have a peptide, we have a complete integrity of the membrane of the MERSA that's gram-positive, E. coli gram-negative. In the presence of the peptide, we can see that the membrane fluidity has been changed, and you can see membrane degradation of the peptide for both gram-negative and gram-positive. In terms of the stability of this peptide, we did the plasma stability, and if you look at the cyclic peptide like HC and 9C, you can see that they are stable up to 80% even after 2 hours, and then after 24 hours, they're still stable 64% or 70%. But linear peptides are less stable, and they can decompose quickly. And this is normal because linear peptides are more sensitive to degradation, and they don't have the rigidity that cyclic peptide. In terms of the structure study, we did the NMR study of the peptide 8L, and as you know, the environment, lipid environment changed the chemical shape and also position of the peak. And then if the interaction of the lipid, for example, lipid that mimic the bacterial membrane with the peptide happen, then we have a high hydrogen bonding or hydrophobic interaction. We have a broadening of the peak. So when we don't have the lipid, the peak look like this. But then as we increase the lipid, we can see that the peak are broadened and completely becoming flat. So this shows that actually this arginine residue have initial contact with the lipid and generate hydrogen bonding, and then the diphenylalanine generate hydrophobic interaction, and we can see the broadening of the peak. We have the similar NMR data for the cyclic peptide. In terms of the computational study that was done by my collaborator, Roman, in Moscow, he did the molecular simulation of this peptide with the bacterial membrane lipid. And then if the peptide is cyclic compared to the linear one, we see that the peptide initially have initial contact with the phosphate blue group in the membrane. And we have the arginine residue bond here. And then we have an up conformation and down conformation that the peptide penetrate to the membrane, and this hydrophobic residue interact with the hydrophobic chain. In terms of the linear peptide, we have much less penetration compared to cyclic peptide, and that explains why this cyclic peptide have much higher activity, antibacterial activity. So this molecular modeling study actually shows that the cyclic peptide generate more deep penetration in the hydrophobic chain of the membrane in the bacteria. And he did another study that look at the complementary hydrophobic, hydrophilic potential, and look at the peptides that are linear and cyclic. And in the presence of the membrane and interaction with the hydrophobic and hydrophilic residue of the membrane, we look at the matching. In terms of the linear peptide, we don't see much matching, complementary matching. But in terms of the cyclic peptide, we see a huge amount of matching with the complementary amino acid in the hydrophobic region. And for example, 9C and 8C, as you can see, we have much higher hydrophobic potential. In terms of cytotoxicity of this peptide, I selected one peptide, for example, R5W4, and we tested them against different cancer cell line and normal cell line. Normal cell line, we don't see much toxicity for many of these, up to 100 or 50 micromolar. So the peptide actually are not toxic against the normal cell line. In terms of the combination therapy, if we combine this peptide with different antibiotics, we see synergistic activity against gram-negative and gram-positive. And this graph shows summary of some of the data. For example, if you use R5W4 with topramycin, you see that we have a FIC index that shows the synergistic, usually 0.5 or less than 0.5 show the synergistic activity. And again, this peptide have a bactericidal activity, and similar data I showed you before, and it also caused the leakage of the membrane. And if we look at the electron microscopy, R5W4 also caused the membrane denaturation for both gram-positive and gram-negative. And if we look at the resistance, if we do the bacterial cell culture and do multiple passage, eventually the bacteria should show resistance to antibiotic. And we see this for ciprofloxacin, but in terms of the daptomycin and our peptide, we don't see any resistance to bacteria even after 18 passage. Animal study actually showed that the peptides are not toxic. So we use histology and use different mice at different concentration. And if we compare this peptide with vancomycin, if we don't have any compound, most all the mice die after 3 days. But in the presence of 0.2 milligram of the compound, we have 50% survival even after 3 days and after 6 days. Vancomycin was used at higher concentration, shows the similar activity. So we have a very good survival study. And in terms of the toxicity and loss of body weight, we can see that the peptide actually don't cause body weight, and all the animals gain their body weight after 5-6 days. So in conclusion, we have designed broad spectrum antibacterial peptide, and that they have activity against multi-drug resistant. Animal and molecular dynamics stimulation showed that we have peptide interaction with lipid, and we had a significant synergistic activity and low toxicity, and we have also efficacy in vivo. I would like to thank Dr. Efremov and Dr. Konshina from Moscow Academy of Science for doing the modeling, and Dr. Masleniko for animal study, and my collaborator, postdoc, and student, and funding from funding agency. I'd be happy to answer any question you may have. Thank you so much for your attention.
Thank you very much for the nice talk.